Out so much. Our first speaker is sick, actually, and emailed us yesterday. So Mark Davis is one of the really great immunologists, and I'm a poor second to to back him up. But since I drew the short straw to give the lecture, and since I have taught immunology at Princeton University for many years, it isn't so hard for me to sort of come in and say a few words, but it won't be in the detail that Mark would normally have given it. And the good news is the second and third lecturer are here in the room, and we're not going to let them out. And so we're going to be okay. So let me begin by making an evolutionary argument. I mean, it actually gives me an opportunity to insert a whole section on developmental biology, which you normally wouldn't have gotten, but on the developmental biology of the mammalian hematopoietic system, which actually has a lot of lessons that harken back to the things we heard about in the first 4 days in the course. So let me start with evolutionary biology. It's not obvious from anything you've learned. In fact, it's not obvious from anything most people learn. That there's a very big difference in developmental biology of the development of organisms that have passed the threshold evolutionarily of having a backbone, the vertebrates. Right? Vertebrates are something called an amphioxus or a fish. These are animals in the water, and then fish are vertebrates, and then amphibians are the next group up, and reptiles are the next group up. And then reptiles split off into birds and to mammals. And that group is really significantly different in an evolutionary sense. It has a totally different strategy of life, actually, and development. The development of the organism has a totally different strategy than the invertebrates, which don't have a backbone. They may have an exoskeleton, but they don't have a backbone. So let me start with that, because it's fundamental to understanding why the immune system, the mature immune system, really developed in vertebrates. There's an immature immune system that developed in invertebrates. All right. So let's, classical invertebrates are flies that you heard about yesterday, and worms, which you haven't heard about. The strategy, what happened in evolution is that evolution starts with lots of single-cell organisms, at least two kinds of bacteria, the archaea and true bacteria, the eubacteria. And these single-cell organisms most frequently divide by binary fission, and the two offspring look a lot like the mother, except they're half the size, and then they grow to a certain level, and then they divide again, right? So they're in rather constant division. What's quite interesting is next week, as we hear about aging, I don't think you'll hear much about it, but bacteria actually age. So there's a mother and there's an offspring, and the offspring lives longer than the mother will live. And that's true as you move. There's a sort of nice intermediate stage in bacteria whereby they're single cells and they're swimming around, and then some stress or some urge to mate, the hormonal urge to mate occurs. And then they all swarm together and they form a multicellular organism, right? So this sort of intermediate in evolution of multicellular organisms that formed, right, comes from originally attraction between individuals that were separate individuals. And then they do an interesting thing. They actually differentiate and change their shape and everything. So there's an intermediate group called the slime molds, which are in fact amoeba. They go around, they eat bacteria. When they run out of food, they're starving, they all come together and they form a stalk like that, right? And so all the amoeba are lined up in a stalk, and they're really a very different tissue type almost, right? And then the stalk forms spores on top. And the spores, they go from diploid, right? Because they've mated, to haploid, that's meiosis, right? To a single copy of chromosomes. And the spores fall off. The spores are for bad times, right? They're also for mating, but they're for bad times. And then when bacteria come back in, the spores germinate and they're amoeba again, and then they eat again, and then they go over and over again. And that was the first trial at getting a multicellular organisms in evolution. And it's kind of interesting. It also introduces very active sex types. There's actually in some slime molds like Dictyostelium 3 sexes. And then in others, there's 2 sexes and so forth and so on. Then right around 600 million years ago, or maybe 700 million years ago, some of you have heard of the Burgess Shale, but that's just one place around the world where a whole new set of processes in evolution came in, quite interesting processes. Actually, a gene which was a transcription factor, right, and which encoded the ability to make a cell type, right, got duplicated, and then duplicated again, and then duplicated again, right? And so instead of a single cell, you got organisms that looked like that, like centipedes, right? And they were just repeating cells, right? And these duplications were just repeating cells, and they became centipedes because there were 100 of them, and then they had appendages, and they all had the exact same appendage, so they all had 2 legs from back to front. And the only thing that was really different was that they had a head and they had a tail, and the head they took food in, and the tail was their anus, and they took out waste material, right? So the strategy of building organisms started with the duplication of these transcription factors. Just for technical terms, let me just say these are called homeotic genes, and everybody has them. You have about 20 different homeotic genes. And then it wasn't hard to think about going from worms to flies or centipedes to flies. All you had to do was you made a head, you made a thorax, you made an abdomen, right, and you made a tail. So in other words, there were a bunch of cells in the head, there were a bunch of cells in the thorax, there were a bunch of cells. And the head, instead of having the feet, had antenna. So now the transcription factors are actually programmed to make a head with an antenna, right? And the antenna hook up with the nervous system. And the abdomen is programmed not to make, it's programmed to make some feet, but it's also programmed to make wings, right? So these all come from each other, and they're just we're all segmented essentially. You're segmented, right? You've lost your tail, but other than that, you're fine. Everybody has this segmented kind of process. And then the strategy in these invertebrates, so that's the explosion of different animal types, is with these homeotic genes. Because now you could imagine all the, whether you had 2 sets of wings or 4 sets of wings, or whether you had a whole bunch of things. And again, as you heard, evolution is about predation. It's about sex, how often you can mate, how often you get resources and predation, right? And survivals and so forth. And all these invertebrates had the same strategy to develop. Now development becomes a big deal because you have to make a head, you have to make a thorax, you have to make an abdomen and so forth, tail. And the way the strategy was, you heard actually, he just referred to it in an indirect way. You have a fertilized egg. The fertilized egg makes a blastula, which is just a ball of cells that are very undifferentiated. They undergo gastrulation, that is to say, the ends of the ball invaginate, and they make a head and tail. They then commit to polarity. They make head, tail, posterior at the bottom, anterior at the top. So you get a polarity in it, and then you start building the organism. And in invertebrates, you build the organism by different larval forms, right? So when you see a moth, it's come from a worm, right? And a worm is a larval form that eats voraciously and gets it to a fly, or gets it to a moth, or gets it, right? So the invertebrates go through these stages, literally, of building up to make a mature animal. And the most interesting thing about the mature animal is it has only one function, and that is to create offspring, to have sex, and to leave the offspring, right? And the mature animal, the worm, the fly, is post-mitotic when it's born. It doesn't divide ever again, right? So That's its fate. Its fate is to be post-mitotic, and the only cells that divide are the germ cells, and another kind of cell which we'll learn about has a legacy in vertebrates called a macrophage. And that macrophage is their immune system, and it runs around eating bacteria and eating foreign organisms and things, and helps them keep the environment clean, right? But there's no cells that divide in the animal, and because there's no cells that divide, to go back to yesterday's lectures, there's actually no cancers that occur in the animal because everything's post-mitotic, right? It's like the retina I told you about yesterday. The retina stops dividing at 7 years, and so retinoblastoma only exists in children from 0 to 7 years. And there's no more retinoblastoma at all because it's post-mitotic, right?
So no stem cells.
That's correct. So the division is all about stem cells. That's this whole 10 minutes of lecture go to exactly where we're going, right? So there is a kind of cancer that occurs in the instars, the larval forms. Why? Because the larval forms are growing. You're making a worm, the worm is morphing into a fly, and so forth and so on. But the strategy is really quite clear. You're going through stages, you're building, you go post-mitotic, and the only thing you're caring about at that point is how many sperm can I make, how many eggs can I make, how much sex can I have, and how many offspring can I have, right? Evolution cares, right? And you do need resources for all that, so you have to continue to eat and things like that. And that's the strategy right up to the first vertebrate, right? And then the strategy changes dramatically. Oh, and so let me give you a little bit about lifespan. So if you're an adult, we sort of, lifespan is, of course, both your early development in utero, your early development outside of utero, and your adulthood, right? Sexually active adulthood. And if you're an invertebrate, right, your lifespan as an adult is short. So on average, flies will live about 25 days. On average, worms will live about 30 days, right? So it depends on, some of the environmental conditions and things, but their lifespan is really pretty short. They're not rejuvenating themselves at all. They are post-mitotic, right? And actually, if you want to know what flies and worms die of, not many people want to know that, but if you do want to know what flies and worms die of, it's mostly actually infectious diseases. So they actually sort of, if you look at them, they look old. The worms look crooked, and they're not eating a lot, and they're not moving very much, and they really just look old. If you look in the microscope and you have a feeling for someone who looks old, that's what the worm looks like, right? And flies are pretty much the same, right? And they're getting overwhelmed by infectious diseases, right? So maybe you'd say their immune system is declining or something like that, or decaying. All right. Now, that's quite a different philosophy of building an organism than a vertebrate, right? First of all, yes?
What does the immune decay look like
in a worm?
In
The macrophages are not functioning really well, but it's the bacteria it seems like the bacteria are taking over in the organism, and you can see them invading tissue, you can see them. So I would say it's failure of macrophages. There's an innate immune system, which you know about, and all of the chemicals that are, beaten up on them, and the macrophages, and that's their whole immune system. It's not much better than that. They do have an avoidance mechanism. There's a just sort of really cute experiment that shows that if you put if you have a worm in a petri dish, you put a predator in, all the worms immediately are alarmed about the predator. They'll all go to one side, they'll huddle together, right? Like fish do in a school or something, right? And all of that is their nervous system sensing the smell of the predator, right? And so they do have avoidance of predators and things, but that's one worm killing another worm, essentially, predator. All right, so in fact, it's quite interesting. So the neurotransmitter for the worm is serotonin. Serotonin is the neurotransmitter for you. it usually is associated with anxiety. And if you make too much serotonin and you're very anxious, a doctor will give you Prozac, right? And what Prozac is a reuptake inhibitor. It actually changes the concentration of serotonin that's taken up again, inhibits the reuptake of serotonin and lowers the concentration. And your anxiety level usually goes down. What's really great is you could give the worm Prozac and its anxiety level goes down. And it just seems to ignore the fact that there's a predator in the room, right? And so that's a good example of evolutionary conservation, right? Yeah. So you can actually discover drugs through worms and things. All right, so our strategy is really quite different. First of all, when the invertebrates start developing, right, they actually can rejuvenate themselves in many ways. So they, for example, if you don't know this normally because you are not little boys, if you cut a tail of a fish off, right, then it will regenerate a tail. If you take the leg of an amphibian off, it'll regenerate a leg. Right? If you cut it in half, actually, in some cases, it will regenerate a head. I mean, a thorax and an abdomen, a thorax, and a head, right? Or that regenerates a tail if you take the other end, and so forth. So there are these very interesting regenerative capabilities that are in the early, we don't have that, but in the early vertebrates, there's a regeneration. And these are all tagged because in all the vertebrates, there are these things called stem cells. And stem cells are fundamental cells that can divide and differentiate into organs, right? Now, there are different kinds of stem cells. So let me just define them, and then we'll just focus on one, right? There's something called a totipotent stem cell. Totipotent would be it makes every tissue of the body, and a fertilized egg is a great example of a totipotent stem cell. It's going to make every tissue of the body. It's going to make all of you, right? It turns out when you make a blastula, right, that's this ball of about 1,000 cells that come from the egg, the fertilized egg. There are a dozen cells in the blastula, just under the dorsal region of the blastula. There are about a dozen cells which are totipotent and make the whole embryo. The rest of it is making all of the membranes and everything. But there are a dozen cells that are going to make the whole embryo. I could take one out, put it in another egg, and I'll have that genotype in every tissue of the mouse. That's how I know in mouse experiments. So those are called totipotent stem cells. Then totipotent stem cells sort of don't just go to differentiated cells. They go through intermediates, which we'll call progenitors. All right? So you go from a stem cell that has all the capabilities to a progenitor which is narrowing the capabilities. And remember I told you about ectoderm, endoderm, and mesoderm yesterday. Ectoderm makes the skin, it makes the neurons. Mesoderm makes your blood. That's what we're going to focus on today. Makes your blood, and mesoderm makes some of your muscles. And endoderm, which makes everything up the middle, right? Your prostate, your ovary, your colon, your small intestine, your stomach, your esophagus, your lungs, right? Everything up the middle and deep, right? So your endoderm is this whole part, right? Those are progenitors in the sense that they are differentiating into defined cell types in development, right? And then it turns out for you, we know most about you, but everything I say about you is true for mice because we know second most about mice and things, right? There are tissue-specific stem cells. And they stay with you for life, right? So in your skin, you have hair follicles. And in the hair follicle, there is a stem cell that will regenerate your skin. So a burn patient loses a lot of skin. They take the stem cell out, and they grow up 10 to the 9th cells in petri dishes of your skin, and then they patch it back on, right? That's the kind of capability these stem cells have, right? So burn patients are treated with their own skin, well, as long as they have some skin that is functional, right? So skin has stem cells. In fact, every tissue has stem cells. And we're going to call those tissue-specific stem cells. And tissue-specific stem cells are with you for life, and they allow you to regenerate yourself, right? And you, as a consequence, have a very long lifespan, right? I mean, people in the early 20th century maybe died at 35 or 40 years old, but that wasn't the lifespan of humans. They died of infectious diseases, like the worms do and the flies do. But once we get past the infectious disease issues, and once we get past the cardiovascular issues, and once we get past the cancer issues, we don't exactly know what your lifespan will be, but it's certainly right now about 80 for women and about 75 for men. Right? That was the numbers that came off one of the slides yesterday when we were looking at it. Right? And it's probably a little longer right now. Remember, lifespan is done by cohorts. So where's our youngest student in the class? I have lost them. Yes, there you are. Yes.
This out yesterday, though.
Oh, you did?
Yeah, I'm not the correct solution.
You're not the youngest student in the class. Stay young, right? So he's in a cohort with most of the graduate students, let's say, and then another cohort of postdocs and another cohort of assistant professors and then associate professors and then emeritus professors and then Clyde and I. Right? So I don't even have Clyde to kick around anymore, actually, after this. I better get to the lecture pretty quick. Anyhow, tissue-specific stem cells are really interesting because they're the target cell of cancer. So everything I told you about mutations yesterday happened in tissue-specific stem cells, and that's why cancers are tissue-specific. That's why you really have to know the tissue of the cancer. And some oncogenes and some tumor suppressor genes cause cancer in one tissue-specific stem cell and not another. Right? And that's this benign versus malignant that was brought up. Right? So tissue-specific stem cells, first of all, are the targets of cancer. So they harken back to yesterday. That's why I've taken you through this whole odyssey to get you to get the feeling. But tissue-specific stem cells also are the way in which you build organs, right? And regenerate organs. So your liver will regenerate. If I take out 3/4, not me personally, but if a surgeon takes out 3/4 of your liver, right, it will regenerate back. And you have two kinds of stem cells in your liver. And they will regenerate your whole liver, which is really quite remarkable. Now, you know already you can't regenerate a heart, you can't regenerate. So it's not so universal. There are ends of this kind of thing. But you can regenerate some heart muscle, and you can regenerate some other kinds of tissues partly and so forth. Kidney, you can regenerate a little bit and so forth. But literally every one of your organs has a stem, right? And those are often called stem cells, right? Tissue-specific stem cells, right? Now, all of your blood is made starts off by a single stem cell, right? And your blood is made up of maybe 27 different cell types, right? So that gives you the range immediately of, now we're going to focus on the immune system. So all of your blood is made up of, starts from one stem cell, right? And you spend your whole life regenerating it, right? So today, just to give each of you an idea of how hard you're working, today, everyone in this room will regenerate about 1 billion cells, right? 1 billion cells you'll make today, right? And you'll have 1 billion cells die, and so the net will be equal, right? So you are turning over, skin turns over, your gut turns over at alarming rates. Your gut will turn over in 4 days. Your skin will turn over in 27 days, right? And your stem cells will keep you going, right? Now you can calculate how many times your gut turns over in a lifetime, and you get, maybe you get that 70 generation limit. I don't know. That'd be an interesting thing to calculate. Yeah.
What is the proportion of stem cells per tissue?
What?
What is the proportion of stem cells per tissue?
In a tissue, yes. So it's different for different tissues. So so if you count the hair follicles on your arm, that's the number of stem cells you'll have, times the number of cells that are stem cells in the hair follicle, right? I'm going to take you through how stem cells divide, and they don't divide in the intuitive way you'd think they would, right? So they're kind of interesting. In fact, the stem cells are really a whole other kind of cell. Don't even think of them as your normal differentiated cell, right? They're a whole other kind of cell, right? Invertebrates don't get cancer. Vertebrates get cancer. You have stem cells, they don't. That's the answer, right there, right? Okay, so the stem cell I'm going to focus on is called your hematopoietic stem cell, right? Hemato, blood, right? Poietic, forming stem cell, right? And it starts off, right? The first time it starts to exist as a defined cell that you can see and define is in the fetal liver, right? So the fetus, while you're being carried by your mother, right? You're a fetus and you're starting to make liver, and the liver has the yolk sac of the egg. That means all of the nutrients from the egg. And in the yolk sac of the fetal liver, right, is a cell that arises which makes blood. So the fetuses and the embryos making their own blood, right? That's the first time that happens, right? It's quite amazing that the mother's not rejecting that fetus, but there is a barrier that prevents rejection and an immunosuppression in a pregnant mother, right? So what's happening here is very interesting. Your stem cells are starting in the fetal liver. And as you develop bones, your stem cells go to your bone marrow. Bone marrow, so your bones are hollow, and they have cells in the middle of it, and that's called marrow. And one part of the cells are in the marrow. There's a lot of support cells, but one part of the cell in the marrow are hematopoietic stem cells, right? Now, we often classify cells by what proteins are on their surface, right? And we call proteins on their surface cell determinants, or CD, right? So now we're going to do hematopoiesis. We have a hematopoietic stem cell, and it has on its surface a molecule which we've given a number, CD34, right? So whenever I say CD34 cells, you will now immediately know that's the stem cells in your bone marrow. That's how I recognize them. I go to your bone marrow, I take out some cells, I put them on a slide, I stain them with an antibody. I mean, right? The antibody has a fluorescent marker, and I could count the number of stem cells you have in your bone marrow, right? So those are CD34 stem cells. I know for sure if I take a single CD34 cell and I put it in a petri dish, now it's going to be a clone of cells, it will make 27 different tissue types if I put the right hormones in. Everything's about hormonal regulation, right? So a CD34 stem cell is your stem cell, right? And it's going to make many different cell and tissue types, right? And it's going to go through progenitors, right? Is this a good time to do this? Yeah. So let me say, okay. So let me first start off. CD34 stem cells, of course, start in your fetal liver. They replicate up, right? They go to your bone marrow. They're fine. You could very much ask the question that was asked. That's why I better answer it. How many stem cells do we have in our body? Is it that when we're born, are there no more stem cells made, so that the count is on for 70 generations and then we die because we make no more blood, or what? What is happening to these stem cells, right? And the answer is these stem cells divide, and when they divide, you have two choices if you want to think about it. They could divide asymmetrically, where one progeny is a stem cell and the other progeny is a red blood cell. It's going to go to become one of your red blood cells, right? That's asymmetric division. But they don't do that. They divide symmetrically. Good news, that means you can regenerate your stem cells. And you can do it now, right? All of you are young enough to have good regenerative stem cells. So you're regenerating, among the cells you're doing, is you're regenerating your stem cells, right? And with time, you'll build up a lot of stem cells, right? All of us will build up different amounts of stem cells. In the bone marrow. In the bone marrow, yeah. Not on the bone marrow, in the bone marrow, yeah. Will we generate a lot of stem cells, right? And the question, how many stem cells do we have, is different in every person. And it's different in every person because now start to think about Darwinian evolution. When I differentiate a cell, it's terminal. It's not going to divide anymore. There's no more cancers. It doesn't matter how many oncogenes and tumor suppressor genes it has. It can't divide, right? However, a stem cell divides for your life, right? If it didn't, you'd be gone, right? So stem cells also are competing with each other for space in your bone marrow. Just like you're competing for space in this classroom, just like you're competing for mates, just like you're competing for resources, right? It's natural selection in the bone marrow, and they are accumulating mutations, and sometimes they get oncogene or tumor suppressor gene mutations. And then stem cells can grow better. Why can they grow better? Because they have these oncogenes or tumor suppressor genes lost, right? And so they start to grow. So some of you have mutations in your bone marrow stem cells and have more stem cells than others have, right? Now, remember I said it doesn't take one mutation to cause a cancer. That's like a benign tumor, right? It's not even reading the mutation, right? It doesn't take one mutation to cause cancer. Makes them grow a little bit, but then it's not a cancer, right? You got to get a second mutation, a third mutation, right? So you're building up these mutations over your lifetime, and when you get the right number, you'll get a leukemia, or you'll get a lymphoma, some blood tumor, or an erythroblastoma, a red blood cell tumor, right? You'll get a tumor, right, if you build it up. But if you don't, it's just increasing number of stem cells, right? So not only are you born with a certain number of stem cells, you will die with more stem cells than you're born with. Why? Because all of the stem cells of your body are undergoing a competition. They are undergoing the kind of competition with each other for space and resources, and oncogene mutations drive that. So if I just take your stem cells and sequence them one cell at a time, I will find a bunch of mutations as a function of your age, or as a function of your exposure to mutagens or carcinogens, or your genetic background, your ability to repair DNA, any of these things, right? And I will find that you have actually more stem cells because you have these mutations, and they're accumulating over a lifetime, right? Now, fortunately, you know, two mutations independently happening in the same cell is a very small probability when you have lots of cells, right? So this isn't happening on a daily basis for you, but that's how leukemias and lymphomas arise, and they arise from can arise from a CD34 stem cell or a progenitor stem cell. So let me get to progenitors. The first pathway I'll talk about makes a cell type called the reticulocyte, right, which makes a red blood cell. Right? This is the hemoglobin pathway. So what you're looking at here is the commitment of the CD34 stem cell to a progenitor cell called the reticulocyte. Has a nucleus, and almost the whole every chromosome site is shut down by the transcription factors, and the thing you're making at exorbitant levels is hemoglobin. Hemoglobin holds iron, that's why it's red. The nucleus is lost in that cell type, in that lineage, and you're making a red blood cell that just has a bunch of hemoglobin in it. Its function is to bind oxygen, bring it to your lungs. Oxygen is absolutely essential for you to digest everything, for you to make energy, right? And it will also bind carbon dioxide. It'll bring it back to your lungs, and you'll expel carbon dioxide, right? So you go from oxygen to carbon dioxide, and you do that through glucose, and you're getting ATP, energy, and you're getting nutrients to build things, and you absolutely need oxygen to live, right? And so these are the guys that mediate it. Now, there are hormones. It's called erythropoietin, which when secreted, that's when you make more, right? So there's every one of these pathways that I'm going to tell you are on great homeostatic interactions. They talk to each other. If you don't have enough red blood cells, you'll make erythropoietin and you'll make more, right? Long distance runners for the Olympics inject themselves, or bike riders inject themselves with erythropoietin. They make more red blood cells, they go farther, they get more oxygen, right? And so now they're drug tested, right? as you drug test these guys, the first thing you find out is that some people inherit higher levels of erythropoietin and have naturally more red blood cells and are naturally better Olympic bikers. Right? They don't have to take anything, right? And that brings up an ethical question about whether you let them in the games or what. You know. So there are, this is so has big social issues, but nonetheless, that's a first good pathway, right? Is the erythroid pathway. A second pathway makes an intermediate called the megakaryocyte. Right? Right? And this, the hormone is thrombopoietin, right? Erythropoietin, thrombopoietin. A megakaryocyte, mega big, karyocyte nucleus, very big nucleus. Then the nucleus fragments all over the place, actually gets thrown out in many cases, and lots of small cells get made, and those small cells are called platelets. And platelets are the surface of cells on which your blood will clot. Right? So you cut yourself. Right? What's important is to get the bleeding stopped pretty quickly, and you have a rather elaborate, which is not made by this system, made by the liver, a rather elaborate clotting set of agents. But clots have to form upon surfaces, and you've cut yourself and you've lost the surface. And these platelets all come to those surfaces, and they secrete hormones, the platelets, which are epidermal growth factors. They are factors that make your skin grow. And they stop the bleeding right away, right? Because the clotting is occurring on the surfaces. And they insist that your skin stem cells will grow, and you'll grow it in. And if you're young, you'll heal in a day or two, and you'll never even see a scar. And if you're old, you'll heal in a week or so, and you'll see a scar, which tells you something about stem cells as you age, right? Okay, so platelets are a second kind of thing. And again, these all hormonally regulated. Now we get to the first kind of cell. Now, almost every other cell has something to do with the immune system. So I'm going to get to the first class of cells that has to do with the immune system. These are big time players called myeloid cells. Myeloid cells make many cell types. So it's a progenitor that goes in many directions and many cell types. So let me tell you a little bit about these cells. One class of myeloid cells produce granulocytes. All right? Granulocytes are phagocytes. They have granules in them. That's why they're called granulocytes. These are cells that you could just cut yourself, look in a microscope, and you'll see these cells. Right? So granulocytes are pretty common. They're in your, right? They have lots of granules. In the granules are lots of proteases, collagenases, a whole bunch of enzymes which are degrading, right? And they will phagocytize bacteria, take them in, and degrade the bacteria. Or they will spit out enzymes that will degrade your tissue and the bacteria and not let an abscess form, for example, by degrading the tissues. Right? So they are protection against bacteria, and they are pretty destructive. When you get a big bacterial infection, you're losing tissue because the granulocytes are attacking yourself as well as the bacteria. All right? All right, so granulocytes are one kind of myeloid cells. Another kind, I just want to make sure I get this all in, yes, are a whole class of cells called macrophages. Phage is a term for eating, right? Macrophages is eating a lot. They also will phagocytize bacteria, take them into vesicles, special vesicles, and degrade the bacteria, right? There are many kinds of macrophages, A, B, and C, and we don't have to worry about them in great detail. But macrophages actually do a number of things. They go to the liver, and they make a cell that, so many macrophages are in your blood. In fact, many macrophages are also in your, you're going to find that there are lots of organs that store these cells. One is your spleen, and macrophages are in your spleen. Another set of organs are called lymph nodes, which are everywhere. Here, you know, when your lymph nodes swell, you'll feel it. They're hard. Those are the lymph nodes, right? Macrophages are there, right? But they also, in some classes of macrophages go to the liver, where they're called Kupffer cells, and the liver has phagocytic activity, right? So if you have an infection in the body, the liver is helping you phagocytize, and those are Kupffer cells, right? And then some go to the brain. This is part of the immune system of the brain. They're called oligodendroglia, right? You don't have to worry about these things. Oligodendroglia are cells in the brain that phagocytize bacterial infections or any particle. By the way, they're independent of in phagocytosis about doing any particle will be phagocytized. But a bacteria is a good sized particle. It's about a micron. Right? And so you have brain macrophages and you have liver macrophages, and your macrophages in your lymph nodes and in your spleen, and then they're in your blood, right? And they're just cruising around looking for a whole variety of bacteria. This lineage is the old lineage that was in flies and worms and so forth, and was helping protect the flies and worms. And then there are macrophages that produce dendritic cells. And dendritic cells actually present foreign substances to the immune system, which are the lymphoid cells, and I haven't talked about those yet. But the presentation of foreign material is done on a cell, right? What's foreign material? Viruses, bacteria. So when they digest the bacteria, they get it out on a surface receptor, and they present it literally to the immune system and say, this is a foreign protein, make something that'll kill it, right? Spread this systemically instead of just in a cell. And dendritic cells are the terrific cells for presentation. So dendritic cells are presenting cells for the immune system. And they're all made from the macrophages. So look at how many intermediates, the progenitor intermediates there are so far in macrophages. And they're finally, a final kind of myeloid cell is called a neutrophil. This is just like a granulocyte, but it doesn't have granules, but it's phagocytic. So this whole class is phagocytic, and it's populating your body with all these different cell types. And of course, they all look different. They're all differentiated. Everything I'm telling you, we'll find out next week, is regulated by epigenetics, right? The genome's the same in every one of these cell types. And what's different is the transcriptional programs. And the transcriptional programs are regulated by something called epigenetic changes, which really is just acetylations, methylations, phosphorylations, protein modifications of histones, which are wrapping which the DNA wraps around. Okay? All right, so that's the myeloid components. Myeloid components most recently have really come into their fore. Let me just tell you that at the age of 60, okay, so again, there's not a lot of us here at the age of 60, but at the age of 60, 1 in 10 people have elevated myeloid cells. What do I mean by elevated myeloid cells? That when I count them in a blood sample, the numbers are high, right? 5-fold high, 10-fold high, 20-fold high. But it's not a leukemia or lymphoma, it's benign. It does have mutations. It has oncogene and tumor suppressor gene mutations in those myeloid cells, in those progenitor cells. And it's age dependent, the age of 60. It's also dependent on carcinogens and mutagens. If you've been exirradiated in the past, you'll have it at an earlier age. If you've been given chemotherapy, you'll have it at an earlier age. If you smoke, you'll have it at an earlier age, right? At the age of 70 to 80, 7 in 10 people have elevated myeloid cells. What are elevated myeloid cells? They are progenitor cells that have oncogenes and tumor suppressor genes active in them. And they're expanding the population, but they're benign tumors. They're not going to kill you. At least we don't think they kill you, right? That's all you can say at this point. You got to just kind of watch people. They can go with another mutation or two to a disease called acute myelogenous leukemia, right? They can go to that. They are a precursor of leukemias, but they're benign tumors, and they're filled with these mutations, right? That's a great example of stem cells competing with each other. And what are they doing? They're not differentiating well. All the selection pressures for evolution is for division and numbers, right? It's not for differentiation. So all the mutations they accumulate and keep are how many cells I can make, not how well differentiated they are. So you're losing differentiated functions in the myeloid compartment because of that. And that's called clonal. They're clones because they come from a single progenitor cell with mutations in them. It's called clonal hematopoiesis, right? And you see that with everything. You're going to see that with B cells and T cells and so forth. Almost every one of these guys, because of the stem cells competing for cancers, competing for growth. They're very Darwinian, right? Our stem cells are the equivalent of the Darwinian populations in us rather than in the population, right? You getting this? Yes? Oh, okay. I keep looking at you because I keep being enigmatically, can't figure out whether you get it or not. I like to look at the audience and see if they get it or not. And you've defeated me in in figuring out whether you're getting it or not. But you're smiling. That's a good sign. Okay, so that's the myeloid cells, right? I could probably pull in another one called the basophil. Many of these cells are named for how they stain, right? So this will stain very blue and so forth. Basophils actually are cells that first get put in the blood and then they settle in the skin. They're all over your body and settle in the skin. And in the skin, the basophils will make histamine. All right? So when you have a basophil in your skin, right, and you're allergic to something, right, so there's a class of antibody I will tell you about called IgE. It's just a particular protein that's an antibody. An antibody has a receptor that binds to an antigen, right? That would be pollen, right? Whatever. Something you ate, whatever. And then it has on the other end of it a way to attach to a basophil. And it does that. It attaches to the basophil with the antigen, and the basophil throws out the histamine, and histamine contracts your bronchioles. Can't breathe, right? Contracts your bronchioles, contracts your blood vessels. Get cold, right? Blood vessels not going out, right? And you have an allergic attack. In the worst case, you undergo anaphylaxis. If there's too much, anaphylaxis is you can't breathe at all, and you'll go to the hospital and get an injection of epinephrine, or you'll have an EpiPen that'll block the histamine from working. It'll dilate your blood vessels. It'll dilate your bronchioles, right? So these are the cells that are, histamine mediates these allergic reactions that come from the immune system that I haven't really got to yet. Okay, I probably could do one more here, right? There's something called eosinophils. These are all staining components, and eosinophils make a number of chemicals which treat parasites. Right? So if you have parasitic worms, if you have parasitic amoeba, it's the eosinophils that go after it. They pour a lot of crap out. You feel like hell. The worms don't like it. You don't like it. But you get rid of the worms usually, right? So eosinophils have been developed mainly to sort of hit the parasites. They probably do a number of other things that either I don't know or nobody knows. I don't know. But I certainly don't know. All right. And then there is this wonderful group of cells that are the topic. I've killed an hour, and they're the topic of the whole conversation. They are lymphoid cells. And lymphoid cells arise in the bone marrow. That's where the stem cells arise. Yes, so these are the progenitors of the CD34 cells produce a cell called the lymphoid cell. It sits in the bone marrow. If it stays in the bone marrow, it produces a lymphocyte, right, which we call a B cell. And we call it a B cell because it's a bone marrow-derived cell, right? But some of the lymphoid cells will go to the thymus. Thymus is a little organ that you have here. The thymus is a little bit of a peculiar organ. You're born with a rather big thymus, and that's when most of this is happening, that when the T cells go to the thymus. And thymus will be with you until you're, it'll be big until you're maybe 5 or 6. It'll be smaller and smaller and smaller, and by the time you're 20, you practically can't find it, but different people lose it at different rates. Right? And the lymphoid cells that develop in the thymus are called T cells. Right. And these are the serious immune cells that you're going to hear about this afternoon. All right? So let me just start to tell you a little bit about B cells and T cells. Let me say that everything you see above, and the blackboard above, are things that are comparable to what can happen in invertebrates. And many of the proteins that pour out of these cells are found in invertebrates. And they give rise to something called the innate immune system. And the innate immune system is an inherited component of the immune system. Everyone gets the same stuff. And it protects us, right? But vertebrates, starting in fish, have completely developed a new immune system based on these stem cells. And that's in the bottom board. They're the lymphoid cells and the T and the B cells. Right? Okay, so let's start with B cells. They're pretty easy. You'll recognize a B cell right away because on the surface of the B cell will be a molecule called an antibody. So B cells have as their receptor a molecule called antibody. And antibodies are molecules which you'll find in your blood. They're actually secreted. By the way, when B cells activate, they activate by activation, I mean they divide. They activate by having an antigen. An antigen is just a foreign substance recognized by a B cell, in this case, an antibody. Right? It will bind, and this will result in cell division. Right? And it will expand the number terrifically. When this antigen binds, the number expands, and they're all B cells, and then they differentiate into plasma cells. This is really a whole lecture on definitions. Plasma cells, and plasma cells secrete antibodies. So antibodies are secreted, they're in your blood, and they have receptors, and the receptors are pretty specific, right? The receptors are quite remarkable. Then they become nanomolar in their binding constants to antigens, right? So they're pretty remarkable that they increase their affinity with time, you'll see, and how they do that in a minute, right? And they are antibodies are really good at neutralizing viruses in your blood. They're really good at binding to bacteria and getting macrophages to come around them and eat the bacteria. They are a whole armamentarium, and there are estimated, 10 to the 8th, 10 to the 9th different kinds of B cells in your body. That's a lot, 10 to the 8th, 10 to the 9th. You couldn't possibly have the genetic information for that kind of diversity in your body, and that's why it's called the adaptive immune system, not the innate immune system, right? Okay, so we'll get to how they generate diversity. But number one, antibodies generate terrific diversity. They generate high affinity. They bind to a specific protein called an antigen, right? And even better, right, they have memory.
It doesn't have to be a protein, right?
That doesn't have to, yes. Doesn't have to be a protein. They'll bind to a carbohydrate. They'll bind to polyglucose, for example. It could be a carbohydrate, it could be a fatty acid, could be any chemical substance which is foreign. But its definition is to be foreign. It recognizes self from non-self, right? That's one property of B cells. That's quite a nice property, right? Generates tremendous specificity, right? Specificity increases, binding constant increases with time. All these things sound like magic, right? Right? There's 10 to the 8th, 10 to the 9th different specificities, and it has memory. Now, what do I mean by having memory? It means the first time you see an antigen, that's your first shot of a vaccine, right? It'll take about a week to make an antibody. The second time you see an antigen, it takes about 3 days to make the antibody, and the response is about 10-fold higher. Quicker and higher, right? Memory. And it's specific. In poliovirus, neutralization of poliovirus, there are 3 kinds of poliovirus. Why? Because they have 3 different kinds of antigen, that's all. 3 different proteins, right? If you're type 1 poliovirus immune, you're not type 2 or 3. Right? If you had seen type 1 before but not 2 or 3, you'll have memory only to type 1. Specificity, right?
Immunized against all 3.
We are immunized against all 3. That's why, in fact, you get more than 1 shot, because they interfere with each other if you give them all at once. Yeah. Yeah.
What did the check? So is it the same with B cell, with when the antigen is present, the antibody B cell divides, and then when plasma cells will produce more antibodies, or is it the antibody?
I'm sorry, what? So start again.
Antibody or the B cell divides when the antigen is recognized?
That's correct. Not the antibody divides. The antibody is a protein. The cell divides. But the cell is making a specific antibody.
Okay.
All right? So let me just say, I didn't say it, but it should be clear. When B cells make antibodies, they make 1 antibody specifically, right? So you have then, if there's 10 to the 9th specificities, you have 10 to the 9th B cells, right? Because that's the different specificities. But they're at different concentrations because of your history. And since this is an adaptive immune response, and we come from 24 different countries, it's really quite clear that we have very different immune responses, and we have very different adaptive immune responses in our bodies, right? Just from the organisms we've been exposed to and what we're exposed to and so forth. So everybody, this is really individualized, right? Okay, so they have specificity, they have memory, right? What else? They get better with time. By better meaning increased nanomolar, so forth. Okay? All right. So let's just talk about how you generate this, because T cells are very similar. They do things a little bit differently, but they generate it the same way. All right? It turns out, it wouldn't surprise the physicists for sure. Yeah.
1 nanomolar is sort of 1 thing per cubic micron, roughly speaking.
Yeah, maybe. Yeah. I mean, first of all, there are some that are micromolar and there are some that are sub-nanomolar. But actually, the binding constant gets better with time because it's edited by the terminal transferase, and it's all, you know. Yes, that's right. Yeah, somatic hypermutation is what makes it better with time, and it gets better because the antigen concentration is falling while that's happening. Yeah. So, all right. So, as you might guess, yes.
Yes. These the T cells have like all these specificity and make antigens to men, but then they like normally plasma cells.
Yes, they make. It's the plasma cells that are making literally 1 million molecules a second. So they're pouring into your blood the antibody. It's the B cells have it on their surface. They're not making a lot that goes in, right? But it's the plasma cells. Plasma cell comes from a differentiated B cell, and its job is to gear up, make antibody, and secrete it out into the blood. Yeah.
Do these B cells differentiate asymmetrically?
Yes, they do, in the sense that there's something called a memory B cell. So in other words, you wouldn't remember the whole experience. Like I've said, it has memory, right? Unless you pull off at least 1 B cell and put it in the memory compartment, right? So there is at least some asymmetry in the memory cell. So 1 cell goes to memory and the other cell makes a plasma cell. But that doesn't happen every time. Most of them make plasma cells. And the memory compartment's pretty small. It can be pretty small because these guys, actually one of the things that's great about these guys is they can divide every 6 hours. They are serious players when they see an antigen. Dividing every 6 hours is really a lot. So it's quite an amazing group. All right, so as you might guess, if you wanted an adaptive immune response, right, what you need to have is combinatorics, right? So it turns out that the antibody molecules, let me just draw the antibody molecules. They're 4 chains of protein. They have evolved in such a way, so they have the 4 chains of protein, these are called the heavy chain, and these are called the light chain. The 2 heavy chains that are identical, there's 2 light chains that are identical. I can divide the molecules up into 4 by the heavy chain is 2 halves, right? And every, there's about 50 amino acids in every one of these divisions, right? And these are called domains, these divisions. They fold up and they make structures and then they have functions, right? And so there are 4 domains to antibody, and they have evolved from a single molecule that was once 50 amino acids long into one that's now got heavy chains and light chains. So they've evolved from these guys, right? And sitting here are the antigen binding sites. Sitting here are the sites that attach this to cells. Remember I said that an antibody will coat a bacteria, and then it will take the bacteria to a macrophage, bind to the bacteria, bind to the macrophage, and that's called, this is called the so-called FC site, right? That where this binds to the macrophage, and that kills the bacteria, right? So all these have different functions. This piece has a function about whether it passes the placenta or not, right? Passing the placenta means that your baby's protected by that antibody, right? So let me just say there are 5 classes of antibodies. They're called IgG. Well, there are many more than 5 classes of antibodies. There's 4 or 5 different IgGs. So that's called immunoglobulin, right? We've heard that word before, immunoglobulin. It's the gamma globulin shots you get, right? Then there is an IgA antibody, which is secreted through membranes. So your gut is filled with IgA, your blood is filled with IgG. IgE antibodies are the ones that elaborate histamine, right? So they all have different functions. They're really pretty good at lots of diverse functions. You can see this, why there's 27 different classes of these proteins and stuff. All right, so these antibodies are such that here's the spots which bind to antigen, and antigen you're binding to it, and the goal is, of course, to bind to any new protein that ever arises, any new virus. Remember the Ebola virus thing is like a new virus that arose, right? And we have to make antibodies against it, right? So it turns out we have multiple genes called V genes, V1, 2, 3, 4, 5, 6. Maybe we have 100 of them, 200 of them. And these V genes are just going to encode the information for this piece here, right? Just the V variable, right? Genes, right? And then there is another set of genes. This is all on one chromosome. Another set of genes called D genes, diversity genes. 1, 2, 3, 4, and 5, right? Sorry, these are called J genes, J1, 1, 2, 3, 4, 5. These are joining genes. And these are diversity genes. Right. And here's the DNA, the structure of the DNA, and it's enormous, right? It's like a whole, big part of a chromosome, right? So in other words, the whole thing exists in parts, and you're going to assemble the parts now, right? And you can assemble in many combinations. So I could make an antibody that would be V2, J2, D4, right? Or I could make an antibody that would be V1, J1, or J4, D1, right? Or on and on and on, right? And so I could assemble a million antibodies or so from just the genetic information that I have in parts, right? And that's done by internal recombinations between these genes. Yeah.
Sorry, all the J's in the same cell, or you just pick one?
Yeah, exactly. You're just picking one. Yeah.
Picking one and that's one.
That's right. Yeah, yeah.
Chinese restaurant.
Yeah, so the chromosome would look like this. Right? And I picked V1, J2, D3, right? And I throw away everything in the middle, right? Now I got it, and now it goes into the B cell, right? So the B cell is an unusual cell. It's the one cell of your body that doesn't have your original genome because it's been edited, right? It's been edited to make one protein, one good protein, right? So if I try to make a whole organism from your B cell, you would have one immune cell in the body, right? It'd all be fixed. It'd be too late, right? So this is, in general, how you can generate diversity, but it doesn't stop there, right? There's a second step in generating diversity. Once I have a particular sequence here, right? I have it binding to the antigen at that point. It's just random binding and different trials and errors, but it binds, right? And with time, the antigen-antibody complexes are cleared. They're cleared in the urine, they're cleared in the feces, right? They're cleared by being taken up in cells and being degraded to amino acids, but they're cleared. So the antigen concentration starts to decline. So the binding constant starts to get stretched, and you'd stop, right? And then there is an enzyme called terminal transferase, which is an enzyme with, if I break the DNA, I could add A's, T's, G's, or C's. And I add them randomly, right? And so some of those, I have millions of these antibodies by this time, some of those actually bind better. And the ones that bind better then trigger, again, another cell division, right? And they trigger another cell division, they expand it, right? And then the antigen level goes down again, right? And then then I keep editing, right? And so if you can edit as the antigen concentration falls, the binding constant will go up. That's being selected for. Or go down, as the case may be, if it's a binding constant, right? So in this way, you have memory, and at the same time, you leave the memory with a very high affinity antibody, right? So you're editing the affinity, right? So there's not only a VDJ rejoining step, there's an editing step, right? Okay, so let me just, yeah.
So given that the number of possible combinations is finite.
Yeah, so it is finite. This is finite because the number of genes is finite. And that, let's say, roughly gives you 1 million possibilities. That's not enough. But then the editing gives you many more possibilities. Yes.
Because I was thinking that at some point
The editing is quasi-random. Yeah, yeah.
Just a small comment.
Yes. I'm being.
There is, so when those things are cut and joined, there are nucleotides cut from the end of the gene and nucleotides inserted at the junction, and that creates the largest part of this diversity, not just the gene combinatorics. And then on top of this, you have
The insertions. Yeah. Yes.
So,
Thank you.
this library of genes, so it's variable, right? The more you get exposed to things, more combinations are getting added on, more elements are getting added onto that? Or is there-
No, we all have about the same number of V genes, and they're related to each other, but they may not be identical. They have a lot of polymorphisms, right? So you may be able to do some things I can't, or I may be able to do some things you can't. Just like life, right?
Memory associated with that? Let's say if I'm exposed to something at 5, and then I haven't seen it for a while, just forget and drop it out of the...
Yes. So this. So what happens is, if you see something the first time, it forces a replication up, right? And then with time, it falls and falls and falls, and it goes into something called a memory compartment. I'm being vague because I have no idea what the memory compartment is, to be honest with you, right?
It's a fraction of the blood cells.
It's a fraction of the blood cells, yes, I presume. Since I've never looked at the memory compartment, or I don't know anybody who really sort of says, here's how many memory cells you have or something. So they're different cells, though they're B cells that are different, but they're called memory cells. And when an antigen comes back in the second time, it triggers those guys. And as they replicate every 6 hours, they come back fast. Yeah. So there's a really quite a nice system. Yes?
What changes in the cells that makes them memory cells? Like they express new proteins?
Undoubtedly. Undoubtedly. So the cell has a choice. It's making a commitment, is do I become a plasma cell and spend my whole life secreting antibody and then dying, right? And that's what plasma cells do. Or do I go a different direction? So there's a fork in the road where an epigenetic decision is made, right? I am going to do this, therefore I'm making new proteins, I'm doing these things. If I want to go back, I mean, to say where Kurt wants to put the memory cells, they belong in the lymph nodes, in the spleen, they're just waiting there for you, right? They're just sitting there waiting for you. I'm going to make a cell type that then homes back to the spleen or homes back to this. And homing is an interesting thing because it means you have a receptor on the surface that when the blood goes through the organ, it sticks, right? And so I'm homing back. I could get a receptor that makes me stick, things like that. So it's a different transcriptional program that says memory or says death. All right, so I'm gonna leave it up to Holland. I'm gonna leave it up to Holland to tell you about the T cell system, if that's okay. You have no choice. What is he going to say, right? Right? Yes. Sorry.
Tell us about T cell help.
Oh yeah. So there are two kinds of T cells. Let me tell you. Yeah. Well, so the T cells are recognized by cell determinants, CD3. So if you're a CD3 cell, you're a T cell, right? Just like if you're a CD34 cell, you're a stem cell, you're a T cell. And they differentiated into two kinds of cells, CD4 and CD8. CD4 you may recognize as HIV's target cell. So the reason HIV wipes out the immune system is the CD4 cell is a helper cell. And by help it means this thing. CD4 cells have receptors, right? And when those receptors are triggered along with the B cell receptors, right, they actually make hormones. So they supply hormones, and those hormones actually trigger the VDJ rejoining process. They move the antibody from an IgD to an IgD to an IgG to an IgE and A. So they actually are triggering the movements of the DNA, right? And at the same time, they're helping the production of antibody itself. That's why they're called helper cells. So you don't make good antibody without CD4 cells. And you don't make good T cells without CD4 cells. That's the CD8 T cells. CD8 cells are called killers. And Holland will tell you why they're called killers. But they are a very important part of the immune system.
You can probably guess that.
There'll be a test on why they're called killers, yes. But basically the processes are the same. If Holland botches the whole thing up, just think of it as the same, right? Okay, this is probably a good, well, let's ask if there are any questions, because I'm not an immunologist. Yes, yeah.
The VGJE combination only affecting this first domain?
It's affecting, yes, only in here, right, where the bindings are. Those are called, these are called variable regions, of course, and there are V regions here and here, right? And then there's this called hypervariable regions where the editing occurs, which is, as Victor says, the great majority of the diversity is the editing.
Do most edits end up making nonsense?
Yes, so 2/3 make nonsense. And the reason 2/3 make nonsense is because it's a triplet code. And if you're out of reading frame, you have a frame shift mutation. And so those will get thrown away. And 1/3 are good. Absolutely right. Yes.
Semi-short amino acid sequences that
Yeah, semi-short, yeah.
and
What's the average for T cells? 7 or 8? How many amino acids are added, or how many nucleotides are added in the editing?
5, 6. But I mean, it happens twice because
Happens twice, yes, because there's 2 regions.
What sort of things make the process of like predicting or sort of designing sensitivities from like genes such a challenge? I've heard some people talk about how this is like a very practical.
You mean how do you? Do you mean how do you tell what the antigen looks like if you know the sequence of the receptor? And the answer is we don't. But that's the problem, Victor. It's a good problem. That's a real problem. That's a real problem. I mean, how do you predict what the antigen is when you have the sequence of the receptor? That's a problem Holland's working on, problem Victor's working on. So you know there's a couple of people working on that problem. Everybody, sorry. Everyone's working on that, thank you, that problem. I didn't throw the truck.
the antibodies, they become, they go, I don't know where they start, IG.
They start out with IgD first, then they go to IgG, E, yes, so they go to A, then E, yeah.
So
There's a sequential, yes.
DNA somewhere away from this BBJ region where
So this is this part, right? But everything from here on in is called the constant region. This is called the variable region, right? For obvious reasons. The variable region is where the antigen receptors are, right? The constant region is what gives it its flavor of antibody. By flavor, I mean if I'm an IgA antibody, I'm a secretory antibody. If I'm an IgD antibody, I pass the placenta. If I'm an IgE antibody, I trigger histamine release, right? So different antibodies have different functional consequences for the body. Everybody with allergy has high IgE in allergy season. Well, not everybody with allergy, but everyone who has that kind of allergy. Yeah. There are several kinds of allergies. Yeah.
So the choice of the B, the J, and the D,
It's random.
but does it happen when they create a B cell, or does it not?
Yes, so it happens in the bone marrow before, yes. And one of the things that I haven't brought up, but I hope Holland will just address briefly, is for B and T cells, ultimately, you don't want to make autoantibodies, ones that attack yourself. So there has to be a way to eliminate self, right? And that's called tolerance. Now, a lot of tolerance, not all, but much of tolerance is due to a comparison of creating something, testing it against self, and if it is a self, killing it. So that's called clonal elimination, right? Let me just say, every B cell makes one kind of antibody and that's it. Every T cell makes one kind of receptor and that's it, for specificity, right? So it's the ensemble of all the T cells and the ensemble of all the B cells that give you the broad immunity that you have. This is all very clonal in the body. Yeah. I think you understand this. This is hard. Yeah, there are an enormous number of details which I have left out. And Holland will take care of, so I'm not worried. That's, yeah.
some of the things that are not understood, or that you can't explain, are really rather basic.
Yes.
Questions that probably
Give us a couple of examples.
I was thinking, I'm totally confused about how T cell
Helping.
works.
Yes. So T cell help,
But it's
right?
not that they both, but the T cell and the B cell recognize totally different things.
Yes.
And therefore, there's no way the T cell directly recognizes the same thing the B cell recognizes. Okay,
So the T cells have antigens, have receptors, and B cells have receptors. And what Kurt is pointing out. So this is a T cell. It's a CD4 T cell, so it's a helper, right? And this is a B cell. And it binds antigen A. And this binds antigen B, right? But they're in the same vicinity, right? And the binding of antigen B triggers this cell, this T cell, to divide. And in the division process, it secretes a number of hormones. Let me give an example of a hormone it will secrete. It will secrete a hormone called interferon. And interferon will spread to the cells in the vicinity, and it will raise the concentration of certain proteins on the surface of the cell. And among them are called histocompatibility antigens, which you're going to hear about in a few minutes, that make receptors. And those receptors are critical for the binding of these two cell types. And when they are close and they trigger, then you're starting to do things like these hormones helping to get this B cell prepared for a VDJ rejoining, right? Or prepared for moving from an IgE to IgD to an IgG, or in other words, the putting together of the genes for these three domains and this domain, right? They keep switching with time. It's, and you're right, there is the details of these mechanisms have not been studied. Well, actually, you know what? I'll tell you the problem, and then we'll sort of take a break.
I'm on Wikipedia right now.
Google is treacherous. Yes, yeah, yeah. But no, I'll tell you what I think the problem is. It's a very interesting problem. All of these things have been studied by people called immunologists, right? Now, I can say that because we don't have really any card-carrying immunologists in the room, so I'm going to insult them. And they are not trained like molecular biologists. They are not trained like people who worry about signal transduction pathways and a whole bunch of other kinds of things usually. And so they do fundamental things with the genome, but not fundamental things with the signal transduction pathways, which are, in fact, a very important signaling back and forth issue, right? It's just a matter of who is playing in the sandbox together and what they know, right? So that's why a course like this is very good, because we are increasing the diversity of the children playing in the sandbox, right? That's not an insult. So I think that's what's needed for these kinds of fields. People, haven't paid. Usually, people do what we're doing here. You get a lecture on immunology, and that's what you know about immunology, right? And it's just not the details. But there are, buried in all the things I've said, enormous number of problems that are interesting, right? So we've had lecture actually heavily based on development, going into developmental immunology, okay? And Holland will start to ask slightly different, many different questions, yeah. All right, so why don't we take a break?